Okay, sir, am I audible now?
Yes, sir. Yes, sir.
Okay, I think yesterday we already discussed that primary screening and secondary screening. And once you success in the terms of screening of specific microorganism which exhibit some traits, industrial important, biotechnological importance, strengths, trait, then it is considered as a valuable for the mass production of the particular product, particular metabolites. For example, antibiotic production. But sometimes what will be happen, that strain originally you have isolated some natural sources, natural habitats, and they having their own particular capacity to produce the metabolites in the terms of quantity. So if you change that strains through the mutation, through the some other RDNA technology like techniques, then the chances of the production of the particular metabolites what you are desired may be increased with the same setup, with the same particular resources. So that particular part today we will learn, that is improvement of your selected strain, bacteria or the fungi. So the screening for the productive strain and the strain improvement in the biotechnology organisms is very important, because the source of the microorganisms used in biotechnology, that is known well, selects from naturally occurring variants, for example, from the soil, from the water, from the mud, from the marine sources, manipulation of the genome of industrial microorganism in the strain improvement. So that three particular aspect is very important. We will discuss that particular part. So I think screening has been carried out for the number of the different particular metabolites, starting from antibiotic to the uh, toxins or some enzyme inhibitors, some enzymes, organic acid, growth promoter, pigments, and the appropriate screening methods have been already used and we have already discussed yesterday. Okay, sometimes the antibiotic are used. Okay, so that unwanted organism cannot be grown. Sometimes there's some particular inhibitors are used. Okay, so that depends, and you can screen the some different compound which having some certain activity produced by the different particular microorganism. So source of the organism used in biotechnology, you can ultimately either search from the literature, and based on that you can order your particular culture from the culture collection center, and they supply to you if there is a viability of the culture. So this is one. Otherwise, the isolation de novo of organisms producing the metabolites of economic importance, for example, antibiotic or pigments or enzyme. So the enrichment with the substrate utilized by the microorganism and enrichment with toxic analogs of the substrate utilized by the microorganism being sought are the two methods through which actually you can find out the some isolation of those organisms which produce the certain industrial important metabolites, and testing the microbial metabolites for the bioactive compound. Bioactive compound, that is having some particular activity against the some cancer cells, some pathogens. Okay? So they are known as a exhibit the bioactivity. So this is, I think, the literature search and the culture collection center. We already, discussed the different culture collection center available in Gujarat, India, and the third point, from where actually you can take the such particular culture. Isolation of the de novo organism producing the metabolites of economic importance. Although I think that the well-known ubiquity of the microorganism implies that the almost any ecological entity, whether it is the waters or the air or any particular leaves or the tree trunks, may provide the microorganism, because microorganisms are ubiquitous and they are omnipresent. soil is considered as a source of the isolated microorganisms. Okay, and you know that in during your routine practical, I think you at the n number of times you take the soil sample and go through the isolation of fungi, isolation of bacteria, isolation of actinomyces, and blah blah many other particular aspects. So soil is considered as a nothing but the very rich reservoir of the diverse group of microorganisms. So microorganism capable of utilizing virtually any carbon sources. Okay, any carbon sources will be found in the soil if adequate. So am I audible? Hello? Hello?
Yes, sir.
My voice is hello.
Yes, sir. Yes, sir.
Okay, and PPT is also visible, no?
Yes, sir.
Okay. So what we are discussing, that is microorganism which is present in the soil is capable to utilize any carbon sources. Okay, this is found into the nature. So the things is that right now what will be happen, the number of the particular microorganism which is found in different soils has been already investigated, and millions of the different bacterial species have been identified by different microbiologists. Okay, so nowadays few microbiologists or the industrial microbiologists, they are ultimately searching the new habitat like deep sea, I mean marine resources, okay, or marine environment, and from there they are find out the bioactive compound or metabolites, and they may having a, I mean number of the examples in front of us is there where actually the drugs is originally detected from the marine environment, and for that they utilize the specific for the find out the such metabolites which having a biological activity, and for that there is a three-step methods are used. One, that is enrichment with the substrate utilized by the microorganism being sought. Okay, enrichment with toxic analogs of the substrate utilized by the microorganism being sourced, and finally testing the metabolites for the bioactive activity. So enrichment with the substrate utilized by the microorganism, that means you already known that your microorganism is, for example, okay, thermophilic one. Okay, you provide that particular temperature. So what will be happen? Your target organism is, for example, thermophiles. Okay, and you provide that high temperature. So what will be happen? You are one time, you are enriching, okay, enriching the that particular condition. Or for example, your organism is utilized cellulose, and add the cellulose into the that particular site. So only those organisms will be dominated which you able to utilize the substrate. Okay, so this is one. And I think we have already discussed that particular part. Okay, so I'm just omitting this part because this is nothing but the enrichment. Okay, so depending upon the which substrate is available, you can able to ultimately find out those organisms. So enrichment with the toxic analogs of the substrate utilized by the organism being sought. It may be used for the enrichment and the incubated with the soil. So toxic analogs will kill the many organisms which utilize that particular compound, and the survival are then grow onto the medium with the non-toxic substrate. So in such particular condition, you can see that you can ultimately carry out the screening and testing. Testing of the for the bioactivity. Bioactivity, that means you already isolate the certain organism, and after that you culture it into suitable media, and as an extracellular extract you are ultimately tested against the different bacteria, different fungi, or the yeast. They are known as an antimicrobial activity. You can also use that particular bacterial extract for the enzyme inhibition. Enzyme inhibition, that means those enzymes which are therapeutically valuable. Therapeutically valuable, that means that enzymes if you inhibit, then you can treat the disease into the human. testing for the morphological changes into the fungal test organism. Okay, so that means it works against the fungi, and if the morphological changes is found into the fungus. Okay, and then animal testing conducted into the animal, okay, for the microbial metabolites. So these are the, for example, this is testing for the antimicrobial activity. Zone of inhibition will be there. You can find out. Depends on testing against the enzymes. Okay, so enzymes, those enzymes which associate with the certain disease and may lead to be isolated and tested for the ability to produce the drug combating disease. So this is the method which is available for the enzyme inhibition. So that is generally, percentage inhibition is calculated. Okay. This is second method where actually the quantity of the, that unreacted substrate is generally unreacted substrate. That means enzymes not utilize that substrate. Okay. And that can be calculated by C minus A minus, and that is substrate with C minus B to 100. Okay, C is the reaction without the enzymes and without the inhibitor, and from that you can ultimately, this is considered as a normal screening. Okay, where actually qualitative like screening, that is quantitative like screening. Testing. So testing for morphological changes in fungi, because for hyphal morphology may be used to detect the presence of the pharmacology active compounds. Then conducting the test onto the microbial. So what we are discussing, that is nothing but the certain aspect of the screening and how they are ultimately used to find out the particular organism which having some biotechnological important traits or the industry valuable traits, because they are producing the particular metabolites. Now we switch over to the strain improvement. Okay, so why the strain improvement is required? It is required because the efficacy of the production of metabolites will be increased or to be increased. Optimization of the growth and the utilization of the wide variety of the substrate. Okay, so such particular can be modified for the betterment and viability of the process. So in short, the improvement in the terms of growth parameter, in the terms of their efficiency to produce the particular metabolites, or sometimes for the improve the overall economy of the your process. Okay, so that's why the strain improvement is considered as a very key process. So why we are ultimately going to start the strain improvement? What are the some target? First, that is you want to achieve the rapid growth. If the rapid growth is possible, your the production times will be reduced, and if the production time will be reduced, so those product which is produced 72 hours, and you will reduce that particular through the rapid growth within 48 hours, so the times will be saved, electricity, everything will be saved. Okay, so in that sense, it is generally rapid growth. Genetic stability, organisms should not be undergo through the genetic mutation. Non-toxicity to the human, it should not be a pathogenic one. So in that terms, it is non-pathogenic. So large cell size for easy removal from the culture fluids. Removal is very important one in the terms of nothing but to separate your product from the culture, whether it is intracellular or extracellular. Then ability to cheaper, use the cheaper substrate. If your organism is utilized only this, pure source of glucose, and the cost of the glucose for 1 kg, for example, is 2000 rupees. And the same times you utilize the, for example, those particular substrate which is readily available, for example, in dairy waste, lactose. Okay, so which one is good for the, your desired organism? Those which utilize the very cheaper substrates. Okay, elimination of the production of the compound that may interfere into the downstream process. Sometimes what will be happen, your product will be produced, okay, but along with that, sometimes some other particular byproduct which produced by the same organism, okay, and that creates some problems in downstream process. Downstream process, that means after the fermentation, whatever the harvesting, purification, recovery are there, in that particular process, such particular compound may interfere. Okay, so you can eliminate the such particular compound or the product produced by this microorganism through the strain improvement. So I think now you can get the idea why or what is the target for the strain improvement. Okay, you can increase the productivity, improve the use of carbon source and nitrogen source, reduction of the cultivation cost by lower price in nutrient and the requirement of oxygen. Okay, you can also sometimes carry out the strain improvement for the production of some additional enzymes. Your microorganism, for example, already produce the XYZ product, but you just improve the strain, okay, so that apart from your XYZ product, it also produces the ABC product. So that is byproduct or additional product. So ultimately you are targeting. For that, actually strain improvement is possible. See, another one, that is you also modify the microorganism, okay, to produce the inhibitory compound, okay, and that inhibitory compound will inhibit the growth of the or contaminated microorganisms. Okay, so these are some of the target. I think most of you know that. Okay, to change the genetic makeup, to carry out the alteration, okay, in the strains or the species, genetic engineering is considered as a very viable option, where actually the recombinant DNA you produce, and that recombinant DNA technology is useful for the strain improvement. So I'm not going to detail in that particular part because the partially it is generally already covered. So apart from genetic engineering, which is generally used for bacteria and the yeast, parasexual cycle is also used for filamentous fungi, where the parasexualism is generally found. Protoplast fusion, I think many of you know that the two cell is fused, okay, together to produce the one giant cell, which is very easy in the plant biotechnology, generally used for the yeast, fungi, and actinomyces. Transduction and transformation is also conjugation. All are there, but they are not that much used, and it is only restricted to the bacteria only. Okay? Mutation is also there, but the site-directed mutagenesis is generally not that much used. The reason is that there is a random site-directed mutagenesis may sometimes destroy your culture rather than improving the culture. The destroy of culture may ultimately occur. So generally, mutation is considered as a spontaneous or induced one. Okay, and one particular drawback of the mutation is that sometimes you may lose, that your strains or the product of your strains. Okay, that depends. Okay, which process you are going to utilize. Okay, spontaneous mutation and natural mutation. Okay, naturally occurs. Then direct mutation, induced mutation, and the site-directed mutagenesis. So basically, the four different particular, the types of the mutagenesis is there. Okay? So it depends. For example, induced mutation is, you can use because you are inducing the mutation. So for that, actually, you may select some different mutagens like ionizing radiation, like gamma radiation, which cause the single or double stranded DNA back. And the deletion and structural alteration is generally carried out. No doubt it is not that much used. Okay, UV rays and chemicals. Okay, for example, different chemicals are there. UV rays, ultraviolet rays, you can also use that cause the dimerization in the pyrimidine. Okay, then hydroxyl amine, that is actually carried out the deamination of the cytosine. Similarly, nitrous acid is also used, which causes deamination of not only cytosine but in A, C, and G. And biologically, Mutagen like phage, plasmid, transposon are also used. But you have to ultimately select the particular mutagens as per your experiment, as per your requirement. And after that, you may ultimately apply it because they each having their own drawback or the demerits. For example, normal DNA for over here, for example, at the fourth nucleotide, there is some mutation. What will be happen? That mutation leads to the changes into the codon. Okay, UUA is there, but in fourth number alphabet is changed. Okay, instead of A, there is a T is there. So what will be happen? AGT, rather there is a TGT will be there. Okay, so TGT is the codon on mRNA, that means ACA rather than UCA, and so such point mutation may ultimately alter the polypeptide chain. Okay. Similarly, deletion from that particular may ultimately change the entire the subsequent amino acid sequence. Okay. Even similarly, addition is also change the subsequent polypeptide chain. Okay. Why? Because in case of the normal, for example, alteration of the substitution of the one nucleotide, like for example, in 3-number codon ACA is there. Over here, ATA will be there. So ACA, that means on mRNA UGU and UGU for cysteine. But over here, it will be UAU. So it will be tyrosine. So such substitution of the nucleotide may change. I mean, I think this is normal for you and may ultimately having a aware regarding the direct mutation and everything. So in this mutation, physical or the chemical agents are generally known for the, known as a mutagen, and they are generally used to cause the to create the mutation. And thousands of the different particular mutants you can ultimately screen and find out whether the anyone is superior producer or the inferior producer of your product or okay. So it is not to say select them on the basis of 1 or 2 criteria, but superior mutants is selecting using the more than 1 criterion for the formation of your desired product. So selection of the mutant giving the better, primary metabolites. Any metabolites is produced by the particular microorganism by a certain particular metabolic pathway, which is originally from the central metabolic pathway. The concentration of the particular metabolites increase, then the organism having the regulatory, mechanism to control the overproduction of those metabolites. And if by mutation such control is blocked, then the improved product can be produced. For example, citric acid. Citric acid will not be concentrated by the cell because it is the part of the central metabolic pathway. But if there is an enzyme is blocked or the enzyme is mutated, and then that citric acid will not be ultimately channelized for the subsequent other alpha ketoglutamate or the malic acid, and that's why it is concentrated. Okay, so if you are success to cause the mutation into the certain enzymes, then the citric acid will be considered as nothing but the concentrated. So this is the example of similarly what we are discussing the citric acid. There is a I think this is example of Corynebacterium glutamicum, okay, where the the deficient into the production of biotin as well as in synthesis of the alpha ketoglutarate dehydrogenase. So that means you are ultimately searching for those strains which defective in the certain enzymes, and that's why that enzymes is defective. So the whatever the substrate which is utilized by that enzymes will not be ultimately produce the product. So that substrate is concentrated. Yeah. Okay. Similarly, the selection of the mutants producing the improved level of the secondary metabolites. So the design of the producer for the isolation of the mutant overproducing the secondary metabolites is more difficult due to the fact that the far less information is available on the control of production of secondary metabolites than the primary metabolites. So that the end product of the secondary metabolites are not required for the growth, while in case of the primary, they are essential for the growth, and that's why the pathway, the enzymes required for the carry out the different reaction into the pathway, they are already known one. But in case of the secondary metabolites, this is not true, or this is not ultimately correct. So screening is used or achieve considerable success in selecting the mutant for the production of the secondary metabolites. So several workers have obtained the improved secondary metabolite producing strains by the isolating the oxotropic media. In many cases, there is a no correlation between the compound and the secondary metabolite produced. Possible explanation for this may be that double mutant and their oxotropy was not directly related to the improved productivity. Okay. Then side-directed mutagenesis, the outcome of the conventional mutagenesis is the random, or one can say that it is unpredictable. Sometimes you may lose the strain, sometimes the strain is there, but strain lost the traits or the product formation. So in case of site-directed mutagenesis, that means you are targeting the particular site inside that particular gene and alter that gene. Okay, so sometimes what will be happen by altering the particular site into the gene, the product may ultimately having some superior activity. Okay, so mutation using the oligonucleotides. So oligonucleotide, that means nothing but the primer. You are deliberately use those primer which having some point mutation, some alteration, some mismatch, and that mismatch is generally used and amplify that gene, and then the amplified product is generally transfer into the bacteria, and then mutant recovered from that clones. And if you are success to isolate the such clones, then that mutagenesis, site-directed mutagenesis techniques is success. Okay, so sometimes certain predetermined bases are replaced and the new gene is reinserted into the organism. And site-directed mutagenesis creates specific, well-defined mutation at the specific change in the protein product. Okay, and it helps into the industrial production of the enzymes as well as to produce the specific enzymes. Sometimes I think I think many of insulin. Okay, nowadays fast acting insulin is available. Fast acting insulin, that means if you take the insulin, okay, through the intravenous, it will be lower down the glucose, I mean sugar. Okay, but sometimes what will be happen if you to take the sugar metabolism is initiated after 2 to 3 hours once you take the insulin. Okay, but such particular insulin is available within the 5 minutes. Okay, your sugar metabolism will be controlled by the injection or the insertion of the insulin. So they are known as a fast acting insulin. It is not available in nature. It is defined or it is created through the site-directed mutagenesis. Similarly, slow acting insulin is also available into the market. Slow acting, that means sometimes the patient is not a true diabetic, it is pre-diabetic one. Okay, and that sugar's level is not cross that particular boundary, but it is near to the boundary line. So for such patient, if they take the the insulin once while a week, okay, then it is act as a very slowly. Okay, so exert the effect for a longer period of time. So such slow acting or the fast acting insulin is nothing but the product of the site-directed mutagen. I mean, such number of the example are there, but this is considered as some key example. Then parasexual cycle, it is also known as parasexuality, which is the process generally found into the fungi. So it is considered as a non-sexual mechanism for transferring the genetic material without the meiosis or the development of the sexual structure. Okay, so a parasexual cycle is initiated by the fusion of the hyphae, anastomosis. That process is known as a anastomosis. Hyphae, that means 2 different hyphae, aerial hyphae, during which the nuclear and the cytoplasmic component occupy the same cell, that is known as heterokaryosis, and followed by the plasmogamy. So mitotic crossing over and the haploidization is the then occurs. So that means the entire the parasexual cycle is starting with the fusion of the hyphae, followed by heterokaryosis and plasmogamy, and then there is a mitotic crossing over. So fusion of the two different nuclei in the cell of the heterokaryon result into the formation of diploid nucleus. Diploid, that means two, and nucleus, that is known as a karyogram, which is believed to be unstable. Okay, and once it will be unstable, that's why undergo through the segregation by the recombinant involving the mitotic process. So once the diploid will be there, undergo through the segregation, the haploid cell will be, so that is known as haploidization. Okay, and mitotic crossing over can lead to the exchange of the gene onto the chromosome. And while haploidization probably involves the mitotic disjunctions, which randomly reassort the chromosomes and result into the production of the aneuploidy or the haploid cell. So like sexual cycle, parasexual cycle will give you the species the opportunity to recombine the genome and produce a new genotypes in their offspring. So parasexual cycle, that is a natural process. If you, but the thing is that you can also induce that parasexual cycle through some chemicals. So you can see over here, the two nucleotides is come together. Sorry. Okay, haploid karyon will be occurs. I think you know that the same cell having a dark dot and the white dot. Okay, and now there is a diploid cell will be there. Okay, half dark, half light, and that diploid, okay, having a random assortment of the gene is occurs. So gray color will be produced. That is known as a recombinant heterokaryon, and they may having a some superior characteristic compared to the haploid original, that hyphae. Okay. Okay, there is some limitation is there because the parasexual cycles has been demonstrated into the only imperfect fungi, like penicillium group of fungi, Aspergillus group of fungi. Okay? Other fungi it is not accidentally found. The process that is during the process of recombination during the growth of the heterozygous diploids may occur in two stages. One, that is the mitotic crossover, which results in the diploid recombination. Okay, I think we already discussed over here. Diploid heterokaryon are there, and then there will be hybridization to produce the haploid recombinant. In normal mitotic crossing, or what will be happen, the diploid cell, that is the where the chromosome number, that is 2. Okay, I mean 2A, and then chromatid replication is occurs, spindle formation and segregation is generally carried out. So each cell having a enjoying the 2 chromosome. But mitotic crossing over, sometimes what will be happen, the half of the portion of the chromosome may extend to the another particular chromosome. Ideally, it should be this daughter cell contains the 2 white color chromosome and this is 2 dark color chromosome. Okay, but the thing is that during the mitotic crossing over, there is some exchange of the certain particular the chromatic material. Okay, so now that cell, which is generally through the mitotic crossing over, having a few particular fragment that is new. Okay, similarly over here, this the certain particular fragment is new over here. Okay, so that is generally nothing but the mitotic crossing over. Sometimes what will be happen in haploidization, the one daughter cell having a 2n+1, okay, there is a 3 chromosome, and another having a 2n-1. So this is also a abnormal segregation is occurring. So as a result, the such particular supercell or the such particular single chromosome containing cell is also produced. How you can detect the such particular genetic events? Okay, through the auxotropes, through the colony characteristics, or sometimes the abnormal growth of the fungi may lead to the detect the such particular characteristics. Limitation of the parasexual cycle, that is auxotropic marker on the synthesis of the direct product is sometimes difficult to ultimately detect. Second, that is auxotropic mutants have been observed to have a quite unpredictable result with the production of the secondary metabolites. Even conidial color markers have not been shown to, have shown to deleterious effect onto the product synthesis. In short, there is some particular problems into the auxotropic marker as well as sometimes the conidia colors or the abnormal properties. So frequency, I think we already discussed, okay, that frequency of the heterokaryon can be increased by chemical and physical mutagenesis, like X-rays, nitrogen mustard, or fluorophenylalanine and the ultraviolet light. Okay, so mitotic segregation can be induced by the such mutation. Used for the to investigate the genetics of the industrial important fungi, allocate the genes of the interest, producing the metabolites. Gene mapping on the chromosome is also possible. Okay, next one, that is the protoplast fusion. That is nothing but the cell, two cell which ultimately divorce of this cell wall, and in specific isotonic media, you fuse that two particular cells. Okay, then there will be a formation of the protoplast. And that protoplast is nothing but the fusion of the two particular identical cells. And once it will be fused, then again the cell wall will be developed. The process is very easy. You ultimately select the two protoplasts to be fused. Nuclear fusion is occurs in the strands. Fusion protoplast and may regenerate the cell wall and grow like a normal cell. So it is applicable to the actinomycete, fungi, yeast, even plant cell also. Okay. This is example of cephalosporin production. So where actually the recombination was obtained by both sister and divergence process, a cross between an asporulating, slow-growing fungi with the sporulating, fast-growing strain, which only produced the 1/3 of the cephalosporin level of the first strain, eventually resulting to the isolation of recombinant. And that is desirable property, that is nothing but strain having demonstrated the good sporulation with a high growth rate and 40% more antibiotic than higher yielding parents. That means asporulating will be now becomes the sporulating from the second cell. Slow growing is also ultimately become moderate or fast growing by merging of the cell. Okay, so this is one can say that parents A, parent B, and this is the recombined cell or the protoplast fused cell. So you can see over here the penicillin production will be parents A having a 11.7 and 18.5 milligram per centimeter cube, and you can see that this is the 18. Okay, and spore per slant will be also increased. Germination frequency is also nearly 50% because parents having a 99, B having a 4. Okay. Advantage: no need of selection marker, can apply to the most of all the types of the cell, easy to perform. Fused cell can be modified again by mutation. Once you having a fused cell, then that fused cell is again you can select for the other method to modify it, like mutation. Okay, applied for primary metabolites and secondary metabolites products. Okay, RDNA technology. So RDNA technology, where actually you produce the nothing but the some recombined or recombinant vectors. Okay, and that is procedure is nothing but you have to require the DNA which encode the particular product, require the plasmid, or instead of plasmid you can use the pulse that ultimately deliver your desired DNA into the cell. So you also require the host cell. So these are the hardware to carry out the genetic engineering. The vector is plasmid or the phage is required. Foreign genetic information into the vector, that means the particular methods through which actually you can produce the recombinant vector. Okay, and then at last you can also require the the cuttings or the joining of enzymes, DNA enzymes like restriction nuclease, ligase, and finally the host cell. So this is the diagrammatic procedure of genetic engineering. For example, you will have a foreign DNA, cut it, that DNA so that your desired DNA, which is present over here, will be ultimately available in pure forms with some identical ends. Same particular identical ends is found, should be found onto the that RE cutting plasmid vector so that annealing is possible. The sealing of the nick is carried out by the DNA ligands. We will having a recombinant plasmid that to be transferred to the host cell like yeast or the E. coli, okay, and then it is maintained. And you can go through the replication, then you can assay for the product whether a product is produced or not. Okay, commonly used plasmid vector, I think that okay, PBS series of plasmid, PUC series of the plasmid they are used. Okay, having some selectable marker. Okay, so cloning site is also available from where actually you can carry out the cloning into the vectors. Cloning site, that means nothing but the cutting site. Okay, the structural endonuclease identifies a certain site where actually a DNA is inserted. That is known as a cloning site. So production of the number of the proteins like factor 8 is the blood clotting agent used for the treatment of the hemophilia. This is the first particular human source of the therapeutic valuable compound which is produced through the RDNA technology. Insulin, I think that this is the first in the history of genetic engineering. Insulin is the first metabolites produced through the RDNA technology used to control the sugar metabolism, diabetic patient. Growth hormone, tissue plasminogen activator, erythropoietin, okay? Colony stimulating factors, okay? Interferons. They all are ultimately used in different clinical disease, clinical conditions. They are used, but they are produced through the genetically engineered, strain. This is an example of the type of the enzymes which is produced to the genetically engineering, like protease, phytase, pectinase, peptidase, lipase. Okay, they are used at the different particular process at industrial scale. So, use of the RDNA technology for the improvement of the native microbial products. I mean, self-cloning is carried out. Self-cloning, that means the gene is present into the E. coli, cut that gene, paste again into the same particular cell, okay, and then the production will be carried out. Now the question will be generated: if the gene is already present into the E. coli, then why again you have to cut and paste into the same E. coli? Sometimes what will be happen, the gene product which is produced by the E. coli is very low concentration. Why? Because only the one gene is viable. If you cut that particular gene, paste into the vector, and vector is generally inserted into the bacterial cell, same bacterial cell, and that vector is high copy number. For example, maintain a 20 copy number. So that means your DNA to be cloned into the vector is also 20 in number. So the 20 times more production will be found. Okay, so this is nothing but the heart of the self cloning. So the example of the enhancement of the enzyme production by the genetic amplification, okay, with the same particular donor is bacillus, and sometimes the recipient is also bacillus group of organisms. Okay. So we are not discussing that genetic pathway. Okay. This is nothing but the blockage of the certain particular enzymes. Okay. So this is control of aromatic compound, aromatic amino acid family in Escherichia coli. So there is some control is there. Feedback inhibition control is there. If you overcome that particular part, then concentration of the that product will be increased. Okay. Okay, so this is improvement of industrial strain by modifying the property other than the yield of product. You are not concentrating only yield of product, but you are concentrating some other particular parameter of strain. For example, selection of the stable strain, genetically stable strain resist to the phage infection, so it will not be lyse. Selection of the non-forming strain, because the forming is also a very crucial problems into the industrial level, industrial strain. Selection of the strain resistance to the medium component. Selection of the strains morphologically favorable strains. Strains tolerant to the low oxygen tension and the less undesirable product and high in the interest. But then transduction, that is the transfer of bacterial DNA from one bacteria to another by the bacteriophage. Generalized transduction and specialized transduction are there. This method is well-established research tools in bacteria, including actinomycete, but it is not that much particularly valuable or useful into the fungi. But however, transduction is the general, this is generalized process, this is specialized process. In specialized process, where actually the phage particle carry the DNA, bacterial DNA, along with the phage DNA inserted into the bacterial DNA, and then circulation is occurs. That phage DNA contains your desired, that DNA to be cloned. Clone, that means to be transformed. So those parental cell, bacterial cells which do not, exhibit the particular property will be able to exhibit the certain trends after the phage mediated insertion of specific gene. So when the foreign DNA is absorbed by the cell and they integrate within the genome of the donor cell, cell in which the transformation can occur are known as competent cell. And generally such competent cell are genetically transformed cell and the ability to produce the certain particular product. But sometimes what will be happen? The certain particular product which is produced by the transformed cell sometimes degraded inside the cell, will not be transferred at the outside the cell. So this is one drawback also. Conjugation, this is nothing but the conjugation pillars is there. So F plus cell, F plus, that means maleness cell, okay, produce that pillars and transfer the plasmid to the F minus cell, and F minus cell becomes F plus cell. But once that during the transfer of the plasmid, if you having a certain gene onto the plasmid and that plasmid is transferred to the F minus cell, then F minus cell becomes the positive within the terms of nothing but your plasmid containing the genes. Okay, but however, it is not that much useful. Okay, transduction, conjugation, they are not that much particularly useful. So the novel genetic technology right now available are the metabolic engineering and genome shuffling. So in the metabolic engineering, is nothing but it enables the rational designs or redesigning of the metabolic pathways, okay, of the particular organism through the manipulation of the gene, so as to maximize the production of the particular biotechnological products. So in metabolic engineering, whatever the existing pathway are there, it is modified, or sometimes entirely new one is introduced, okay, through the manipulation of the gene, so that to improve the yield of the microbial products, okay, and eliminate or reduce the undesirable side products, so that your organism is just like becomes one factory for the production of the product, okay, and they are only dedicated produce only your desired product, and other side undesired byproduct will not be produced. Okay. So this is a one kind of modern evolution for existing procedure is used to induce over production of the product by blocking this pathways. Exactly, sometimes what will be happen, your product is produced from the particular pathway, and in this particular pathway, whenever the starting of the raw material is there, substrate is there, that substrate is sometimes also channelized for the production of some other byproduct. But if you are not ultimately require that byproduct, then you just block that particular byproduct pathway. So what will be happen? What is the raw material or the substrate which is now available for your product formation pathway? And in this way, you can ultimately carry out the one direction, okay, dedicate direction to the towards those particular reaction where actually your substrate 100% utilized for your product formation. And in such particular site-directed mutagenesis, in such particular case, site-directed mutagenesis is also sometimes used. So application overproduce the amino acid of high solution in Corynebacterium glutamicum and the ethanol by the equally have been employed to induce the gene for utilizing the lactose in corynebacterium glucan, and thus making it possible for the organism to utilize the whey, that is the dairy product based, which is plentiful and cheap available raw material. So through the metabolic engineering, the gene for the utilization of the xylose was introduced into the Klebsiella species, making it possible for the bacteria to utilize the wood sugar, because wood sugar is contains the xylose. So various primary metabolites overproduced by the through the metabolic engineering, what we have discussed, alcohol produced by Gemmomonas mobilis, that now introduced into the E. coli and Klebsiella. Then secondary metabolites, similarly transfer of the gene from streptomycin erythraceus to the streptomycin levendens, facilitate the products of the erythromycin antibiotic. Okay, another one that is a new method, that is DNA shuffling. This is nothing but the one way through which it will rapidly propagate beneficial mutation in the directed evolution experiment. Genome shuffling or DNA shuffling using the recursive genetic recombination analogs to the DNA shuffling offers one advantage to advantage of simultaneously changes the different position throughout the entire genome. So recombination of the genetic traits among multiple initial strains can be completed in the process of genome shuffling. So actually in genome shuffling, this is one concept, and that concept is generally executed by the one method that is known as a CRISPR-Cas9 technology. What is CRISPR-Cas9 technology? That is clustered regulatory interspaced short palindromic repeats that is naturally found into the bacteria. Okay, so that is known as the CRISPR-Cas9 techniques that is used in many organisms as a genome editing tools because of their high efficiency and the easy manipulation of the genome. So shuffling, that is nothing but the you just are doing some DNA experiment. Okay, you copy and paste or changes some genetic makeup for here and there. So that is nothing but the genome shuffling, and that shuffling is possible through the CRISPR-Cas9 technology. So it is applicable for the bacteria, particularly bacillus strands have been attracted significant attention due to the production of the biological product. No doubt, it is not that much easy to carry out the genetic manipulation into the that bacteria. Okay, but however, CRISPR-Cas9 system now is considered as one of the best systems applied for the genetic editing, and it is proven to be very efficient and precise, and through which actually the genome shuffling is generally possible. So this is a diagrammatic representation of the how actually, no doubt it is a very complex procedure, okay, where actually you have to require the guide RNA, then also require the protospacer adjustment motif. Okay, you have to require the double-stranded break causing the enzymes like Cas9 mediated DNA. Cas9 system is generally used for the double-stranded breakage of DNA. Then whatever the particular homologous overlapping sequence that is represent over here in red color, okay, that will be mutated in such a way that there will be insertion or there will be deletion is there, and that insertion and deletion, okay, may cause the the shuffling of the DNA, changes of the DNA. I mean, this is more advanced compared to the site-directed mutagenesis. So this is all about the different techniques which is generally used for the strain information. If you're having any particular doubt, you can ultimately ask me for